Okay, I want to welcome everybody to the NHABON Community of Practice webinar number 6, Operating Imaging FlowCytobots. We chose this topic because there is such extraordinary interest in imaging FlowCytobots, but we want to make sure that this is not meant as an endorsement of IFCBs in particular. There are a number of other instruments that can also do this. And we would be interested in holding webinars on those as well at a later date. But we noticed that there were a lot of proposals coming into various programs that involve imaging FlowCytobots. And so we wanted people to have a realistic view of what it's like to operate an imaging FlowCytobot. Could I have the next slide? Oh, I also want to mention that this will focus primarily on marine systems because that is where most IFCBs are being used. However, I have seen several examples just recently of use in fresh water. So first I want to go over the agenda. We're going to have talks by Mike Brosnahan, Kate Hubbard, and Clarissa Anderson. And then hopefully we'll have time for a Q&A and discussion. And then there'll be a very brief wrap up and a mention of the schedule for future webinars. Could I have the next slide? So just to remind you about what the NHABON Community of Practice is, you are the NHABON Community of Practice, and its purpose is to foster scientific collaboration, information sharing, and other activities leading to the development, operation, and application of the NHABON at local, state, and regional levels. There's a steering committee, and you can see all the people who are on the steering committee. We invite applications from new steering committee members every year. So if you're interested in participating, they provide this group with guidance and suggestions about future things we could do. So going to the next slide. So I want to introduce our speakers very briefly. And they're going to speak in the order of that they're shown here on the slide. Mike Brosnahan is an associate scientist without tenure at Woods Hole Oceanographic Institution. His research interests are HAB ecology and life cycle dynamics, and through his work, he's deeply involved in developing the imaging FlowCytobot phytoplankton sensor, as well as platforms for putting it in interesting places. He is currently leading a team that is working to establish a network of advanced HAB sensors in the Northeast U.S. as part of the NHABON effort. Dr. Kate Hubbard leads the HAB monitoring and research program for the Florida Fish and Wildlife Conservation Commission's Fish and Wildlife Research Institute and directs the FWC Center for Red Tide Research. She works with a broad partner network across the U.S. to utilize, sustain, and advance HAB observations. And finally, we'll hear from Dr. Clarissa Anderson, who's a biological oceanographer with expertise in ecological forecasting and remote sensing. She is now the executive director of the Southern California Coastal Ocean Observing System, SCOOS, at Scripps Institution of Oceanography, where she continues to conduct research on phytoplankton ecology and HABs in coastal California. So if we could have the first speaker, Mike Brosnahan.
Okay, can you hear me? All right, thanks. All right, so I am leading off. So I was tasked with introducing the Imaging FlowCytobot, or IFCB, and hopefully you can see those slides now. So the Imaging FlowCytobot, or IFCB, was invented by Heidi Sosik and Rob Olson about 20 years ago. It's an imaging in flow cytometer, and that's a machine that, in this case, automates light microscopy. This particular sensor was specifically designed for long-term continuous observation of the ocean in situ. And it was successfully commercialized about 10 years ago in collaboration with McLean Research Laboratories. So as part of that commercialization effort, McLean's developed some nice animations, and you're looking at one here that's been edited a bit. This one shows how the instrument works. So it collects a sample of seawater from its exterior with a syringe pump, and then presses that sample in a thin stream through a flow cell. Particles in the seawater sample interact with a laser, and they scatter light or fluoresce. And when that interaction is detected, that triggers a flash lamp to go off just as a particle is passing in front of a microscope objective. And so in this way, the sensor collects grayscale images at rates of up to 14 per second. So you're seeing an example there. And these are highly regular images. The optical system is basically unchanged from the first systems that were deployed. So there's IFCB data going back about 20 years now that's all pretty comparable. So a quick sum up, so these images are highly detailed. They are generally well focused. They have sub-micron resolution, and the sensor really targets cells between 5 and 150 microns long, which is a really nice range for capturing many, but not all, marine HAB species. Again, it's designed for continuous in situ operation down to depths of about 50 meters. That can be deployed for up to several months, and it generally is set up to collect samples every 25 minutes or so. It has several anti-fouling features that enable these long deployments. So the most central is this recirculating sheath flow that's continuously filtered. That provides biofouling protection, but also provides hydrodynamic focusing for consistent sharp imaging. And there's additional routines and reagents deployed in the sensor to clean on a regular basis. The system was really designed for initially for installation at piers and other fixed locations that could provide power and internet connectivity. So the instrument produces up to 5 gigabytes of imagery per day, which is a lot for many like cellular or certainly for satellite-based telemetry. It has an onboard x86 computer that can run either Windows or Debian Linux. And recently, McLean has released a new version that's really expanded the capability of the sensor and also has an API, and that's specifically for the Linux variant of the instrument. So this sensor has really obvious utility for HAB sensing, and that was demonstrated first even pre-commercialization by a team led by Lisa Campbell down at Texas A&M University. They deployed a sensor at a pier in Aransas Pass, which is just outside of, or connects the Gulf of Mexico with Aransas Bay and Corpus Christi Bay. And they've detected several HABs now with this installation. You can see in these two plots of Dinophysis ovum bloom in 2008. In black are all the observations from the IFCB, and then in red are microscope counts that were collected through sort of standard phytoplankton monitoring procedures. You can see the patchiness of these blooms, and that's especially evident in this later bloom in 2009 of Corenia brevis, where the standard microscopy observations entirely missed these pulses of Corenia cells that came through the pass and impacted the region. In our area, we've been using it for over 10 years now. Initially through projects that were really focused on Alexandrium catenella blooms. And I just wanted to show some of this data to really highlight how information rich these images are. So it's not just counting cells, it's also providing a lot of information about what cells may be doing. So we use that to track progression through this organism's life cycle. We've been able to show that in some of our systems, bloom declines are really driven by sexual encystment events. So not necessarily by nutrient limitation or grazing. And the other thing that we can do with the images is we can start to look at size metrics in a really automated, high throughput way. So we can use things like biovolume estimates and cell length estimates to look at things like division rates and again, life cycle dynamics. Really key to all of these analyses is a variety of different data sharing and analysis tools that have been developed as open source projects. The first of these was a MATLAB kit that Heidi released with the instrument that provided a lot of ways to do some image classification and other image analyses. Subsequently, there's been some Python tools that Joe Futrell's led the development of, as well as a web-based data access platform called the IFCB dashboard. Louis Kilfoyle is actually Heidi's son, and he's developed a nice annotation tool that we use, and I'm aware of at least a couple others that are in development. There's a convolutional neural network model development application kit that Sydney Batchelder's developed, and we use extensively in our systems here. And then we've been developing something called the HAB Hub, which is a system for sharing some of these IFCB data products and interactive plots alongside other types of HAB monitoring information. As I said, there's a lot of other open source efforts underway or free resources that are being built. I think Clarissa will talk a little bit about a project called HABDAC that's based out on the West Coast. There's some other ones that are afoot as well. Kind of returning back to our use of the imaging FlowCytobot in development of NHABON. So here in the Northeast, we've had a long-standing problem with Alexandrian catenella blooms. This is the, I'm not sure if you can see my cursor, but it's sort of the furthest left grayscale image you see. It's sort of black cell. These cause paralytic shellfish poisoning, and generally blooms are happening in the spring and summer. But over the last 10 years, we've seen the emergence of several other dinoflagellate species in the spring and summer seasons. Some of these are toxic, other ones are impacting aquaculture and fisheries. And then in the fall and winter, we're seeing increasing incidence of these blooms of Sunicia that produce domoic acid and cause anugic shellfish poisoning. So with this sort of growing complexity and new challenges, there's a real need for enhanced surveillance for these species, especially for the new ones where we don't really understand some of the ecological controls that dictate when the blooms are going to happen. And so we set out to establish a network of imaging FlowCytobots and other sensors around the region. And in our case, we're using a mixture of fixed platforms, so building on some established time series that we've set up and our colleagues have set up here in the region, and then also developing some new platforms that allow us to move sensors to different locations with the seasons. And most recently, working with a company called C-Track to deploy the sensors offshore where some of the largest and sort of most impactful blooms are developing. So for our fixed sites, you can see several of them here. These are generally peers. The Martha's Vineyard Coastal Observatory is this yellow tripod in the lower left. That's the longest running imaging FlowCytobot time series. It was established by Heidi Sosik and Rob Olson, and it goes back to 2006. Most of these are more recent than that, and we've been working to sort of extend the ones that were seasonal. The other thing that we've been working on is co-deploying the imaging FlowCytobot alongside another sensor called the Environmental Sample Processor, or ESP. That's this sort of large can you see in the upper set of images. The IFCB and ESP sensors are really complementary sensors. So the IFCB is again, it's a light microscopy system. It's taking pictures, and oftentimes these images are they're very informative, but they're not necessarily allowing us to resolve fine taxonomy, and that's particularly true with Sunicia cells. So by coupling these instruments together, we're able to take this ESP sensor, which can make measurements of particulate domoic acid, which is the toxin produced by Sunicia, and we can kind of couple these together and sort of use our chemistry and the ESP as judiciously as possible and really target periods when the Sunicia are present. So I'm going to just run through this really fast, but you know, this is an example where back in 2020 we had a bloom along the coast of Maine, and we were detecting some reasonably large Sunicia cells that you see in the pictures in the lower right, and we were able to trigger our ESP and make some measurements of the picograms, estimates of the picograms of domoic acid per cell, which is pretty low initially. But as this bloom evolved, we saw the cells were getting larger, some more extensive chain formation in the imaging FlowCytobot images, and we saw increased DA quotas on a per cell basis. And then interestingly, during the termination of the bloom, our cell abundance went down quite a bit. we saw shorter and shorter chains, but the estimates of toxin per cell went up pretty dramatically. And so we've been continuing to operate our systems this way, trying to develop more data sets to kind of resolve some questions around these kinds of observations, and also to connect our measurements of pseudonutrient abundance and the toxicity with the ESP to actual contamination of shellfish. Sort of another part of this effort has been to start to consider the IFCB as a component within sensing systems. So in some of the inshore study sites that we have, we have been we started out coupling the imaging FlowCytobot to things like profiling CTDs that we were jury rigging together. And this turned out to be a really powerful combination for us because we could actually use the IFCB observations to do like in situ calibrations of less things like fluorometers that don't necessarily resolve species in the water. And as over time, this has evolved to really try to harden the system, and we've most recently been migrating it to something called robot operating system, which is a way to develop modular robotic designs, and this allows the IFCB to interact with other sensors and these winch systems that we've been trying to build at pretty low cost. So for instance, we can do behaviors where we have a CTD attached to the imaging FlowCytobot, and we'll drop it slowly through the water column, detect where the chlorophyll maximum is, and then have the IFCB go back to that depth to suck up its next sample for imaging. So that's one area where we've been doing a lot of development recently, and we hope to release a public version of this PhytoArm code in the next few weeks. We're getting really close. But this is really the basis for both these barge platforms that we've been developing and moving around the region. We had a couple of deployments of these up on the Maine coast now. And then this year, we're really kind of turning our focus most intensely on some test deployments of the C-Track autonomous boat system. So the C-Track really solves one of our problems, which is providing power for continuous operation of the IFCB. It's got about 750 watts of solar generating capacity on board. It's essentially like a floating solar panel. But we still have this issue where we're producing just so much data that we can't really operate the boat outside of cellular networks in the usual way, where we just make the imagery and push it to shore. So what we've been doing as part of this effort is working with McLean to start or to replace the IFCB's computer with an NVIDIA Jetson platform. And we've made a lot of progress on this recently. We actually have an instrument that McLean built with one of these computers in it, and we've just demonstrated onboard classification alongside data acquisition. So we're looking forward to deploying this Jetson-based IFCB on the C-Track in a couple of deployments this coming spring and summer. So just another note about this ROS business and PhytoArm, it's all quite modular. So when we release this code, it's not going to be critical that other users have a Jetson IFCB. They'll be able to deploy all of the software that we have across the IFCB and several other computers in a network quite easily. And this kind of comes back to one of our big sort of aims and sort of directions where we're trying to develop for NHABON, and that's to, again, to collect more data offshore so that we can provide, accurate and timely data in ways that are really accessible, and also ways that can provide the sort of most advanced warning to stakeholders and end users as we can. And that really means getting out to where the blooms are developing. So quick summary, the IFCBs in our region are really well suited for detection and monitoring of the marine species that are impacting our region. And for that reason, it's really emerging as the backbone of our network right now. And if you're interested in this PhytoArm stuff, be on the lookout. We hope to release that in the next few weeks. And lots of people are involved in this work, so I'll stop there.
So next up is Kate Hubbard.
Thanks, Quay. So I will be talking a little bit about the development of our IFCB program in Florida. It is a relatively young program compared to what we just heard about from Mike. We started in 2018, and since then we have hit the ground running. We've really benefited from standing on the shoulders of IFCB giants. Within our own team, we have merged our taxonomy and field teams that include ecologists and phycologists. We've been using IFCBs in a number of different ways: routine sampling, event response, and research. I'll go over some examples, some of the successes and challenges that we've had. And then also, currently we conduct data management ourselves, and we work with HUI for some of the telemeter data. We also work with a pretty broad partner network. And I thought I would focus on Carnea brevis today, since this is our primary organism right now that we're using for IFCB work. In terms of monitoring, we get thousands of samples a year that we look at with light microscopy. So in a sense, our goal is not necessarily to have a bunch of IFCBs in the water for routine monitoring, although that would be great in the future. The blooms tend to be very patchy over space and time, as you can see in this image that I've shown here from the bloom in 2021. And blooms vary in severity quite a bit. Just going back and looking at October over the past several years, we can see that in some years there's barely a bloom at all, and some years there's a bloom extending across almost the entire state of Florida. So trying to understand some of the key bloom dynamics, as well as trying to monitor these events, is a big goal of the Florida HAB Observing Network. So this is something that we've been working on formalizing and will ideally be fitting within the National HAB Observing Network. So again, the IFCBs are a really important part of this, and there are a number of different components. In terms of thinking about some of the key questions that we still have related to Carnea brevis, there's this balance between growth and physical processes. This is true for many other HABs, but Carnea brevis is an incredibly slow grower. We balance that with it also being quite versatile in terms of its thresholds in the environment. We also know that it can sometimes be allelopathic. And so as we start to use the IFCB, we can identify typical cells, but we can also look for signs of dividing cells. In terms of the life cycle, the life cycle of Carnea brevis has not yet been fully resolved. It's not yet clear if there's a resting stage. However, resting stages have been identified for the sibling species Carnea mikimotoi. So this is a big effort, and one of the things that we've seen quite a bit with our IFCB work are these round Carnea brevis cells. Whether these are stress cells and or some sort of life cycle transition into a resting stage is another interesting question that we're trying to work on. And these really tie into thinking about a lot of the different complicated mechanisms underlining bloom dynamics that really help us develop stronger models and forecasts. And so there's a bunch of different things that we can look into specifically. So I thought I would go through some of our successes. We are primarily deploying these either via a submerged deployment at a dock or a pier, or mapping via a flow-through system on a vessel, both large and small. With our dock or pier deployments thus far, we've had over 500 wet days, and we've had submerged deployments in Sarasota Bay and Tampa Bay. These have all been tied to shore power. We've started to integrate water quality sensors like SONS. We have successfully gotten a telemetry system in place, and we also have funded a collaboration with the Florida Gulf Coast University. They also have an IFCB, and they've been processing samples benchtop. And I'll show you a little bit of data on the next slide, but really quickly with our flow through efforts, we have routine surveys that leave from Miami and go up past Tampa Bay, and we've had the IFCB on 21 of those surveys. They happen every other month. But we've also been trying to get these on as many different cruises as we can, so that we have more and more data. And that's been really interesting and beneficial to just trying to map out blooms, especially offshore where we get a lot fewer samples. In terms of some of the data that we've been getting from these submerged deployments, with our Tampa Bay deployment in 2021, we were able to capture a transition from dividing cells into these round cells towards the end of that bloom, right before it really dissipated from the bay. We were also interested that the cells were still dividing during that period. We saw them both, even during the time where we saw quite a few round cells. So it's interesting that that division occurs throughout the bloom. That was something that we didn't really know before. And then in terms of our mapping, we just had a survey come back. This is not the image from that survey, but it was great because while the folks were out on the survey, they were sending IFCB data back to us, and we were able to generate these maps in almost near real time. So those are some of the successes that we've had, and we've also had a fair number of challenges. And I would say just in reaching out to my team with this, the instrument and fleet maintenance and logistics is one of the most challenging things, just making sure that all the instruments are functioning, and making sure that we have extras in case they're not. We also have a lot of logistical considerations beyond the typical troubleshooting. For example, on top of installation, we're concerned about security, sample integrity, and anti-fouling strategy to some extent. Those may go hand in hand for Crania and possibly aphicate dinoflagellates. We found that the cells may in fact be sensitive to copper, and that may be one factor that could help with shifting morphology. So trying to think through that a little bit more has been something that somewhat of a challenge, but also been very interesting along the way. Some of the other challenges that Mike mentioned and the solutions, we're right there with him in terms of power, co-deployed sensors. Another challenge that we've had as a state agency is procurement. All of our procurement is tied to an annual cycle, and so sometimes trying to order something like an IFCB, it doesn't necessarily fit within that window. So it takes a little bit of planning to make that work. Another logistical consideration that I think is really important is making sure that there are staff that can help troubleshoot problems, issues that come up, and making sure that we have ongoing training for those staff, both new IFCB users and the more advanced users. One of the things is we come into this arena fairly late in the game. There are already a lot of developed protocols, and in some cases, the initial methods for, say, image classification have already been updated. And so trying to assess which classification scheme is ultimately best for us. Ideally, we want something stable that we can continue to use into the future. And there's some questions about that come up. Same with data storage and management. And it's really exciting to see these efforts develop at the national level, but then wondering, how do we get involved, integrated, given where we're at in our process? And luckily, there are a lot of great people that are willing to talk about this as we go through. So I will wrap up with just some future goals in terms of our ongoing efforts, divided these into field data and research and monitoring. So we're really interested in trying to integrate vertical profiling. We've seen even in deployments where we were in maybe 6 to 8 feet of water, there's a clear diel signal, and it looks like that's likely related to diel vertical migration. We also would like to be able to look at what's going on subsurface on cruises. Right now, we've only been using flow-through systems. In terms of offshore deployments, these blooms for Crania brevis start offshore at depth. So being able to get a buoy system where we can have an IFCB at depth is a big priority, but trying to get the power set up for that has been somewhat challenging. And then I already mentioned biofouling. We've gone through a lot already in our process. You can see one of the instruments when it comes out of the water over on the right, and they can get pretty funky. We continue to work on data management and communication, both within our existing state structure, but also thinking about how we might need to go beyond that or push the limits. And then in terms of research and monitoring, continuing to use the IFCB, not just in the field, but also in the lab. We've been able to set up a really nice flow-through lab system and do some comparisons of quantification. In general, we found that Crania brevis, for example, is underestimated relative to light microscopy. So trying to figure that out. And then a huge priority is just to try to continue to expand this cooperative IFCB network in Florida and to continue to develop the Florida HABON within the national HABON to work on other HAB issues. And I forgot to mention, but one of the things that was a bit unexpected, we've started to see dinophysis more in some of our installations. So that's a good sort of precursor to setting up a program where we can test more for dinophysis in areas like shellfish harvest areas where that is going to potentially be problematic. So I will stop there.
Okay, thank you very much, Kate. Before I turn it over to Clarissa, I want to remind people to put their questions in the question box, and I don't see any there yet because we won't have a lot of time at the end, so it'd be good if people had questions, they'd start putting them in. Okay, Clarissa, go ahead and take it. You're muted.
Still muted. Okay. Hi. Hopefully you can see my screen. And I'm sorry about the echo. I'm in the middle of moving my office today, so it's completely cleared out. I don't normally live this minimalistic. So we're moving around the coast here. Looks like my face is still on the screen, at least on mine. Hopefully you can see this first slide. I'm going to be discussing what's happening in California with respect to IFCBs. I put this slide here, though, to illustrate a lot like what you've seen with other programs and with Kate's program. There's a lot happening. There's a lot of tools in this toolbox for looking at HABs, and we've been evolving this system for a long time. And this is a lot of people in California who have contributed to all the things I'm going to talk about today. And certainly, I won't dwell on this slide, but we've had weekly sampling. It's a program we call HABmap. We've developed models. We now have a bulletin. We're working on pushing the envelope on a lot of models, and just to then build out how we can really serve stakeholder needs, we've added a full network of IFCBs. And so I'm going to focus just on that box for the rest of the talk. A few years ago, we had a really nice windfall of funding from the Ocean Protection Council here in California to build the system out. This was really spearheaded by Rafe Kudela at UC Santa Cruz, who was building out his own network of IFCBs in the Monterey Bay and San Francisco Bay, and really pioneering a lot of this work in California. And that set the stage for us to expand this program, convince California to allow us to purchase 6 more instruments. And then from there, we have worked with partners in academia and in industry to continue to build that network. And we're at a place now where we can say we have 12 IFCBs that are in rotation, and we have 9 stations, part of this real-time IFCB network, that are meant to all be operational. They are operational, however, it does change through time how many are operating at any given point, but you can see them all here. This covers a vast distance in California, starting with the Humboldt region in the north, down to Scripps Pier in the south. And really, there's a lot of people that are not pictured here, namely, I would say a lot of the technical staff who are really doing the preponderance of the work. And I want to illustrate what is just so cool about this. We can talk about HAB detection, of course, but I think even just from fundamental science perspective, it's good for us to take a step back. You've seen some beautiful images about what we can glean about the life cycles of plankton and blooms and bloom cycles. Another thing to appreciate when you have a network like this that spans such a latitudinal gradient is just to see that even when you take a snapshot, say from the one you see here from November 2022, just how different things look across the entire state of California at any given time. Even in Southern California, we can move from Scripps Pier, where you're seeing predominantly a lingulodinium bloom here, or at least the beginnings of one, move up to Newport and don't really see that, just, not even that far at the coast. It's the next county up in Orange County. And then another interesting illustration is to look at Santa Cruz Municipal Wharf here and the power buoy, which I'm going to show you a little bit more of offshore in Monterey Bay. And just to see what that cross-shore gradient looks like as you move from what looks like a pretty nice mixed assemblage of diatoms and dinoflagellates with some cochleodinium or margolipidinium, you come inshore and you have almost a monospecific Akashiwo bloom. And then up in Cal Poly, Humboldt, same time, mostly diatoms. So it's really phenomenal, and I think there's going to be great power in being able to really use these instruments for a lot of stuff, even, and of course, HAB detection. We have as many deployment types as there are instruments. It seems like we've kind of spanned the gamut. You've seen some really great illustrations of peer and offshore deployments. We're pretty much focused on our fixed site peer deployments where we have co-located instruments as part of SCOOS and SENCU's programs that go back a long way, automated shore stations, other co-located environmental measurements. We also have 2 offshore deployments on buoys, and I will illustrate those in a little bit more in detail. And then here you can see kind of this classic problem that we are having, just like you saw in Florida with biofouling. This has been particularly difficult in Southern California in the last few years. We've also had challenges with sediment clogging our syringes, in part because it seems like there's a lot of fine sediment in the in suspension in Southern California that Northern California sites have not dealt with nearly as much. And I also want to point out that we've acknowledged our funding per part of the funding requirements. We have this sign at Scripps Pier, which acknowledges the program. So I'll point out first for the 2 offshore deployments, the first one is in Monterey Bay. It's this power buoy. This is a buoy that you've probably heard of, the M1 mooring, which is a long-standing ecosystem mooring with really great time series going back 20 plus years. And this power buoy uses wave motion to supply power to that buoy. And it was a good platform to put such a power-hungry instrument. You can see it being deployed here. This takes a really big and very experienced team at MBARI to make this happen with SENCU's guidance and funding. And you can see them here putting it into the hull of the surface buoy. And I have another image here forward. So this is just the diagram of what that buoy looks like vertically. And then just an amuse-bouche, I guess, of the kinds of data that are coming out of it. Patrick Daniel in Rafe Kudella's lab has been working hard on some new deep convolutional neural network classifiers. Here's one that's been trained with 50 different classes from over 70,000 annotated images. This is years of work that's gone back for a while here in the Kudella lab, and I'm not going to dwell on any of these one data sets because I'm going to keep this mostly to an operational talk, but just give you some snippets of the kinds of data that we're starting to put out for each site. The Del Mar mooring was also a big motivator for this work and how we thought about framing the California IFCB network. We would love to build out more offshore buoy deployments. However, I will say that the moorings that we had in place that we could leverage has been a huge boon for this, because we can leverage the funding that goes into supporting these quite expensive and time-intensive efforts like the Del Mar mooring. And it was an NSF project with Heidi Sossik's group and Scripps folks to put an IFCB on this mooring some years ago that did give us the motivation to continue this in an operational setting. And Uwe Sen's mooring has helped support this moving forward, even though we are getting quite a bit of O&M from IUCE and from NCOS through the NHABON project. So here is a picture of the deployment. You can see how it's illustrated in this diagram. We have quite a few solar panels that are generating the energy for this, and here it is attached. It's drawn back about 44 meters down. We're working on getting classifiers running for Southern California, and in this case, we've started with an annotated set of images from the Del Mar mooring that were paired with Northeastern Shelf annotations. So this goes back to that NSF project, bringing in this combination of annotated images and trying to train a CNN that would have skill at the Del Mar mooring. And we're seeing a lot of skills, so we're starting to think about how to apply that to all of the Southern California sites. And then from there, refine and continue to train with new annotations. And this is a huge community effort. In terms of just thinking about moorings for yourself, if you were to go down that road, it's helpful to see some of these successes and metrics. Quite a few images. You can appreciate now how much data comes in. We ratchet that down for these moorings. We don't transmit those data more than a few times a day. Right now it's twice. We've had quite a few challenges, I would say, with access at times when things don't work and it's hard to rally the troops to get out there and deal with the maintenance. We've had humidity increases because of that sediment problem that have led to some pretty dramatic recovery efforts. We did have, because we're in a military area, we had a Navy helicopter damage the modem at one point by flying too low over the buoy. So things like that have happened. We're starting to think about ways to flag samples so that we know early on when we're having failures. And we have a complementary project that's helping a lot with that, which I'm going to get into. Axiom, I keep messing up. Axiom Data Science is collaborating with us and was able to help us stand up a dashboard really fast so that we could see all of these sites. You're seeing 14 types of deployments here because we have a number of cruises that we're putting our IFCBs on to do flow through underway, things like Cal Coffee, the Plumes and Blooms cruise line that's been funded by NASA for years in the Santa Barbara Channel. And so we're able to quickly pull down images from this, look at them, and then bring that system into a larger framework that we're hoping will become a national HAB data assembly center as part of this PCM HAB funded project. And Axiom Data Science is serving as the host and central node for this. We bring in our distributed network of IFCB data in real time into that dashboard, generate all of the files that we can generate, but then think about new products that we might want to create and tertiary products to make it valuable to the community. One of those things would be HAB Hub, as you heard from Mike earlier, where we can think about how we visualize these data as they're coming in real time for managers and pair them with all of the other ancillary data we have at those sites. But also thinking about classifiers and making it more of a community center for scientists and managers to alike to sort of dabble with the data, train their own data sets, grab other classifiers. And so we've thought about this kind of menu of options where we would be adding value to products such that if you were to purchase an IFCB and come into the community now, you could easily stand up a dashboard through this DAC. You could automate a whole number of programs and processes to get you exactly where you want. You could have on-demand classifier application, even grab an extant or existing classifier and then work later to train with your new imagery, rather than trying to do all of this out of the box, moving some of these MATLAB development products into Python environment with folks like Joe Futrell. And so we're trying to make this a very collaborative space where we hope that it could be a DAC, not just for HABs, but certainly for plankton writ large, ecosystem scientists and modelers who could come in and really make use of the data, which we hope will become large. Right now it's focused on California and the Gulf of Maine, but we want to keep expanding this. And so we're building a community of practice around that. We have a transition advisory committee. We're building this out, experts, people from the regional associations. We have a data integration group to help with the very technical components where we're thinking about metadata, standardizing the kinds of files that are coming in and creating a real sort of set of best practices around how to deal with imagery data. So I'll just end with saying that we've had some sort of with collating best practices, making lists of things that we think are helpful for others coming into this space or who have been in this space for a while. We've definitely learned we need the hands-on trainings and refreshers with McLean. Because a lot of this got started during COVID, we did a lot of Zoom training. We have an upcoming in-person training in Santa Cruz next week with 24 of our technical staff and McLean engineers, where we hope to have some good refreshers for those who really never really got that hands-on experience. We've realized, particularly because some of the issues we've had with humidity and syringes and other clogs and accidents, that we need to plan for spare parts, maintenance, and repairs, which, at least for Southern California, is something on order 10% of the cost of the instrument per year. We've learned to dedicate one person to technical maintenance, upkeep, and communicating with McLean, who have been great in helping us all along the way, no matter what happens. And so we're collectively documenting all of this, where we buy things, how do we standardize some of the purchases, and then also just leverage the networks that are out there. And HABDAC hopefully will become one of those things that you can all leverage as you come into this space, and as well as HABHub at HUI. And we're looking for international networks and partnerships to grow this beyond just the national space. So my last slide is just a video which I'll play as we move into the Q&A, showing just a little bit more about the deployment at Del Mar. Thank you. My volume.
Sound effects are interesting.
Sounds.
So while we're waiting for some questions to come in, I wanted to say that many of you don't know that this is the 40th anniversary of the first study of the use of flow cytometry in marine research at a Bermuda Biological Station workshop, which I attended along with Heidi Sosick and Rob Olson. And I suspect they, but I know for sure me, that I am just astonished and amazed at what has happened in the succeeding 40 years and what we're capable of. Right now we have two questions, but I'm having trouble understanding one of them because it got cut off. Oh, there it is. Now I can see it. My work is in freshwater. I'm interested in the use of FlowCytobot technology for freshwater use. What would be the success of the FlowCytobot technology in freshwater? Does one of you want to comment on some of the challenges for using it in freshwater? I don't think anybody's actually done it, but.
So I've done a little bit of freshwater work. I'm not a freshwater specialist by any means. I think one of the challenges for us was just getting up to speed on the taxonomy, the diversity of species that we're seeing, which is entirely different than kind of our standard work. The other issue is that the foci were sometimes different. Like microcystis colonies are great because they're these big globular things that are pretty distinctive and are very much in that range of 5 to 150 microns, although they can be much larger too, and then are excluded from the instrument. So particles or colonies that are larger than 150 microns are generally prevented from being sucked into the instrument because of a filter. So I know it's been used with some success by other groups. It certainly works. There's some things to consider if you're moving a lot between marine and freshwater systems, like in estuaries, but they're not prohibitive by any means. So I hope that's helpful.
Thank you, Mike. I think so. The next question, can you turn your mic off, Mike? Because I hear. Thank you all for the information. We're wondering if anyone is working on a shared classifier that would be available for anyone. With everyone having to build their own classifier, it seems like a huge duplication of effort. Thanks. But I think several of you have pointed out the fact that you're building on other classifiers and that you're sharing classifiers. But would somebody like Clarissa like to comment on that?
Yeah, I started to answer this in the chat just to say, Miranda, I don't know if it helped to hear about the HABDAC development and the fact that this will be an open source tool with what we hope will be quite a few classifiers to choose from and apply to your work, and then look at the validation and dive in deeper. Does that help? I don't know if you're able to even unmute yourself.
So somebody is, Allison Deary is pointing out that she's looking for a postdoc. And so if you're interested in a postdoc, check the questions. Masha, can everybody see the questions?
Maybe only you and I can, Quay.
Yeah, whoever's an organizer can see them.
So if you're interested, get in contact with Allison Deary. It's to use machine learning techniques to analyze IFCB images from the Bering and Chukchi seas. I don't see any more questions. We'll give it a minute.
I had another answer to the last question.
Oh, okay. Yeah, be glad.
I think I'll actually point to one of Mike's slides where while there are a lot of benefits in using classifiers that have already been developed, I think one of the really neat things with the IFCB is that you can expect the unexpected. So you don't necessarily know if you're going to see your HAB of interest, but maybe it's going to have some other things going on. Maybe it will look like it's got viruses or fungi or something else, you know. So I think that that's a really strong motivator to continue to learn how to build the classifiers as well as adapt others.
Well, and I think the original example from Texas where they were looking for Karenia brevis and Dinophysis popped up right before an oyster festival and saved a lot of people from being sick. And that's what sold a lot of people originally on doing this kind of work.
I think the other thing I would just add to that too, just to pile on, is that there is a need for sort of better systems for sharing and attributing all the work that goes into making training sets. So the training sets that sort of underlie the models they're just so precious. And there's a lot of heroes in the community right now that have done a lot of work to annotate images. And for any new user, I suggest you reach out to more experienced users and see if you might be able to access some of their imagery to help you stand up to train your own model that really considers the species that are present in the samples you're analyzing. It doesn't make a lot of sense to have everything that could possibly be there in your classifier, because you just end up generating a lot of false positives unnecessarily.
But contacting other users is probably also a wise thing with regard to deployment, purchasing, all sorts of things, because you can learn from what other people have done.
And I think just to add to what Mike was saying, another advantage we hope of the DAC would be to have access to these annotated images so that you could build your own classifier, like he's saying, with taxa of interest. You don't need to go straight to some pre-made classifier, but to have those libraries at the ready would be pretty invaluable.
So we're now getting some more questions. What are the major issues that arise when installing an IFCB in situ as opposed to a desktop installation? I'm not sure they mean a desktop, but perhaps a peer installation. I'll let all three of you answer that. Who wants to start? I'll let Mike. Oh, Clarissa, you got your mouth open.
My computer's fast. I'm sure we all have millions of things we could say here. I mean, it does cause, there's just a whole new suite of things you've got to do. If you're going to deploy it subsurface, I meant to say this, but we have subsurface fixed sites, we have on deck sites where we're pumping. Just those two things alone are big questions with interesting kind of integration solutions that you'll have to come up with. When you have something on a desktop and you're doing flow through, you're just not dealing with that. You're not dealing with diving and biofouling and pumps and just a whole host of things that you would have to deal with in situ. I know I didn't go to those in detail, but Mike, Kate, do you want to say anything else?
Sure. I guess I'm a big proponent of putting the instruments in the water for a couple reasons. One is that it's really an instrument that is designed for continuous operation in situ, and so that's how you're going to collect the data, where you reveal these really these patterns that you're only going to see with high frequency sampling. So if you're using it as a lab or benchtop system, you're not sort of accessing that potential, and there's probably other solutions for automated imaging that you might consider that might be more or less expensive. The other thing that has come up in some cases for us, particularly in warmer regions, is that when the instruments are in air, there can be a big delta between the internal temperature of the instrument and the water that it's sampling. And so it takes about 20 minutes to analyze a sample, and that can actually lead to bubble formation as your sample degasses. And bubbles are sort of the enemy of good IFCB data. So you can have your system really well calibrated, well aligned, but if you're generating bubbles and they get stuck in the wrong place, you're going to make a lot of blurry images, and that's not the kind of data you want to be producing. So it doesn't always happen, and there's ways to mitigate it if you're really stuck and you have to deploy with the the sensor on a pier. But that's why I have a bias towards putting the instrument in the water. The other thing I don't like is pumps, because it's another thing that can break. So when I have the option, I usually try to just rely on the instrument to suck up its own water. But every site's different. So if you have a site where you have people on site often, and they can sort of keep tabs on your pump, it can certainly work well, and lots of colleagues are using pumps with a lot of success.
And I would just add the data accessibility to that. So if you have an instrument that's set up in a lab or hooked up to Wi-Fi, generally it's pretty easy to get data off of the instrument. But if you have to work through data telemetry, that can just be a challenge, and what works in New England and California may not be accessible in Florida. So just trying to work through some of those struggles as well. I saw that as a big difference.
Another question, does internal biofouling risk increase when the devices are installed in situ? You're muted if you're answering, Clarissa.
I was just thinking about, well, for the internal biofouling, I mean, of course, you have your bleach runs. I think that's what you're talking about, is inside the internal tubing, not the external biofouling. You may have to sometimes increase how often you do those bleach runs when you're in situ. But certainly you're going to have more risk if you're in situ than you would otherwise, I believe. I mean, it just depends on the kind of water you're bringing in too. We've just seen so much variability in that latitudinally in California. That'll depend on how productive your site is as well.
Does anybody else want to comment on that?
Yeah, we've had more issues actually with stuff growing on the outside of the instrument. So, several of our instruments now, we actually just put anti-fouling paint on. So to prevent like seaweeds and things from growing on them and occluding the intake. Generally speaking, yeah, the internal systems are pretty good at preventing fouling. That changes if you have any kind of malfunction or you have a power outage and the system is down for a long time, so you kind of lose that continuous sheath flow. That's when we tend to see things crop up.
So we have one last question, but this is going to be a tough one to answer quickly. So we may not be able to give this in much detail. Has the frequency, spatial distribution, and severity of HAB blooms increased over time in New England, Florida, and California? In a few words, answer that question. Clarissa, I'll start with you.
Yeah, that's that $64,000 question where we're all trying to understand that based on the kinds of sampling programs we've had for decades and how those programs have evolved. Even with, we haven't had this kind of network in place long enough for me to really answer that from the IFCB perspective. I think, the longest running IFCBs are Rafes and Santa Cruz. definitely seen some cyclical changes in plankton. We have recorded sort of secular changes in things like pseudonychia blooms and how toxic they are over time. We're still trying to understand statistically if that's a real increase. Does appear to be, but this is not true for every single HAB, and we have emerging species that are showing up more and more, probably because of climate or changes in circulation. Okay, that's mine.
Mike?
Yeah, I don't think kind of similar. I don't think I can answer the intensification question with a lot of rigor, just because so many of these things are cyclical, and we're still trying to get a handle on those processes. But definitely the HAB issues are diversifying in the Northeast, and that comes with a whole host of challenges that are intensifying the impacts of HABs overall.
I don't have anything further to add other than it really depends on the HAB for Florida.
Okay, hang on a minute, I'm pasting some references for Masha. Our next webinar will be on March 15th, 2023 at 3 to 4 p.m. We haven't decided on the topic yet, so we would certainly welcome input on that. And I think that's all that I was supposed to say at the end here, but to thank our speakers for all the time it takes. Yes, I forgot about this slide. And thank you all for attending. If you want more information, this link here, just Google NHABON or the IUCE Association and you will get to it. And if you have any ideas for for webinars, you can contact me at quay.dorch@noaa.gov.